Hello, welcome to Science Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of June 30th. Let's start. First we will discuss a paper published by Gopinath Surendra in the Cell on the identification of pyruvate dehydrogenase in the nucleus and its function in acetylation of histones. Why is this paper so important? Nuclear acetylation is altered in conditions like cancer, neurodegeneration, and in cardiovascular diseases. How the nucleus generates Acetyl-CoA in metazoan cells is not clear. ATP citrate lyase is the predominant pathway for nuclear Acetyl-CoA generation in mammalian cells. Authors demonstrate the presence of pyruvate dehydrogenase in the nucleus and its role in acetylation of histones. Let's look at the data. Authors demonstrate the presence of PDC complex in the nucleus of various cells using fluorescent probes as shown in this figure, as well as biochemically using immunoblots. They also demonstrate dose-dependent increase in labeled Acetyl-CoA in isolated nuclei and blockage of Acetyl-CoA formation in the siRNA for PDC. What is the function of PDC in the nucleus? Authors show that PDC acetylates histones in the nucleus as shown here using siRNA. They also demonstrate PDC in mitochondria and is regulated by pyruvate dehydrogenase kinase but not in nucleus. Authors synchronize cells to the G1 phase by serum starvation for 24 hours and then introduced serum. They saw increased histone acetylation levels first, followed by cyclin A. They also found increase in nuclear PDC levels during cell cycle progression associated with a parallel decrease in mitochondrial PDC levels. Similar results are also seen in translocation experiments. So PDC translocates from mitochondria to nucleus. What are the factors that affect translocation? Authors looked into EGF, which is required for S-phase entry, and found that EGF increased and EGF inhibitor decreased PDC levels in A549 cells. HSP70 that translocates various proteins found to co-immunoprecipitate with PDC, and the HSP70 inhibitor decreased PDC in the nuclei similar to siRNA treatment. Also, authors show PDC is important for S-phase entry and cell cycle progression. So this is an important discovery that might have implications in several fields and disease conditions. Next, let's look into a paper published by Sami and colleagues in Nature Chemical Biology. Authors demonstrate that autophagy enhances survival of neurons in ALS models. Why is this paper so important? Excessive or mutations in TDP43, a RNA binding protein, has been implicated in ALS and FTD. Role of autophagy in clearing TDP43 is controversial. Authors demonstrate enhancing autophagy improves survival of neurons and astrocytes in ALS models. Automated fluorescence microscopy of primary rodent neurons transfected with wild type and mutated TDP43 is shown in this figure. Risk of cell death as analyzed by hazard ratio increased steadily with levels of wild type and mutated TDP43. Authors also show by using optical pulse technology that mutated protein has lower half-life. To understand the role of autophagy, authors screened millions of compounds and identified 3 autophagy inducers. These compounds lowered both TDP43 and its mutant levels in the neurons as shown here. They also lowered inclusion bodies and cytoplasmic localization of mutated TDP43. Autophagy inducers decrease cell death risk in wild type and mutated TDP expressing primary neurons. Autophagic induction improved survival of TDP43 mutant expressed in human iPSC-derived motor neurons. Astrocytes differentiated from human iPSCs exhibited mutant TDP43 related toxicity that is mitigated by autophagy inducers. So autophagy induction could be used for treatment of ALS. Thank you for watching Science Breakthroughs from Science Mission.